What's up? My name is Rana Kamara, and today I am joined by my classmates Arun, Hiryu, Mardula, and Oleg. We are here to tell you a little bit about genetic material and its vast and storied history.
The history of medicine dates back to ancient times, long before the discovery of genes. The concept of heredity and genes date back to ancient Greece, with philosophers such as Hippocrates theorizing about how similar traits were passed down in families. One such theory suggested that the organs of parents gave off invisible seeds that were combined in the womb of the mother. But the concept of modern genetics only started with Gregor Mendel's work with pea plants in the 1800s. He established his theory of Mendelian inheritance.
The discovery of genetic material and its connection with DNA was obtained through a series of experiments conducted from the 20s to the 50s. People such as Frederick Meister, Frederick Griffith, Oswald T. Avery, Colin M. MacLeod, Mason McCarty, James Watson, and Francis Crick all played crucial roles in discovering this connection and defining the basis of our knowledge on genetic material. In 1839, Meischler isolated a substance he found within a nucleus that he at the time called nuclein. He had unknowingly isolated the substance we now call DNA. Further experiments conducted in 1874 concluded that it was made out of a nucleic acid and protein. In 1928, Griffith performed crucial transformation experiments on bacteria in order to mutate its genetic material and cause a change in the bacteria's lethality. Later, in 1944, Avery, MacLeod, and McCarthy discovered definitive proof that the genetic material that Griffith mutated was DNA, rather than being other materials such as proteins or RNA. In 1953, Watson and Crick then defined the double helix structure of DNA we know today, suggesting a model of DNA replication, later winning them the Nobel Prize in 1962.
One of the first uses of genetic materials was in 1818. It was the first successful blood transfusion between humans, which was monitored by James Blundell. While this worked, in the coming years, people would sometimes die from these transfusions, and many wondered why would this happen, because sometimes it would work and sometimes it would fail. The reason for this would not be found out until 1901. That year, Carl Landsteiner would discover the three blood groups, A, B, and O. Through his research, he figured out these types all had unique features that would sometimes prevent them from interacting with other types of blood. And this would cause your body to reject this when they were getting transfused. For example, if I had type A and let's say type B was something that your body couldn't take, then what would happen was your body would then reject this type B blood by creating antibodies to fight against the incoming blood, which would then cause you to have an infection which could kill you. Thanks to his research, people were able to understand which groups are incompatible with another, and prevent people from dying in the future as they would get the correct type of blood that would work with them. We first began using genetic material for identifying people in the early 20th century, but the first known use of this was in 1892 with a murder case involving Francisca Rojas and her two murdered sons in Argentina. She was found with wounds and claimed that her neighbor, Pedro Velasquez, was the one who committed the crime. When police investigated him, he would not yield. So when they sent investigator Eduardo Alvarez to the scene, he found a bloody fingerprint on the bedroom door, which he then took to another police official, Juan Vudicic, who was at that time working on a system for fingerprint identification and had helped found the world's first fingerprint police bureau a year earlier. With his insight, they were able to figure out that the true culprit was Rojas herself after comparing the fingerprint on the crime scene with hers. She would immediately confess to her crime and was sentenced to life in prison, making this the first time genetic material was ever used in identifying the culprit of a crime. Nowadays, people not only have fingerprint databases and blood types, but they also have DNA records, meaning like in cases such as the Golden State Killer, police are able to track down the true culprit using DNA analysis from prior crime scenes, and then tracking down family members. Though it isn't all, these genetic materials aren't always used in the context of crime. For example, there's CRISPR, which is a creation that allows people to edit DNA strands and change them so that they can remove faulty DNA or correct DNA if needed. For example, if there's a cancer-causing gene in some type of cell, it's possible to use CRISPR to genetically alter this and create stuff that is new.
The exponential advancement in genetic research has led to many biomedical discoveries that has allowed scientists to achieve mind-blowing results. One of the most notable examples of which includes Dolly the sheep. Dolly was born on July 5th, 1996. However, it was no ordinary sheep. It was the first animal to be cloned using adult cells, leading to the breakthrough discovery that using genetics, scientists are able to clone organisms. However, this experiment was a tipping point that set off the fiery debate regarding the morality of genetic experimentation. A quote from the Pew Research Center summarizes this event quite well. Dolly's debut set off a firestorm about both the practical value and ethics of cloning, including the possibility of human cloning. Currently, more than 40 countries, including the UK, France, Germany, and Japan, formally banned human cloning. In other countries, including the US and China, there is no legal prohibition on it. The real question that scientists must ask themselves now is not can they do genetic modification, but should they?